Hi, everyone. Ivan here, one of the founders of Phase Genomics. And today we're doing something a little bit different. I'm sitting down with a longtime colleague and friend, and one of my favorite plant scientists out there, Dr. Alex Harkess from HudsonAlpha. I've known Alex for years. He's been doing some amazing plant research. Some of it he's been doing with us and using our products and things like that. But he also has really cool projects that have to do with education and large campus consortia type projects, and just a lot of really interesting science. And so we wanted to just kind of take some time and talk a little bit about sort of what drives him, what motivates him, what motivates his research, find out a little bit about him as well. So, Alex, welcome. I know we're on Zoom right now. Where do we find you today? Where are you calling from?
Funny story. I'm in Huntsville, Alabama at Hudson Alpha Institute for Biotechnology, where I'm a faculty member, and I am actually standing in a empty laboratory space that is my new company's lab. It's Veil Genomics. We're a long read genotyping scaled solution, effectively making thousands of PacBio libraries and generating insights for people at scale. So I'll give you a quick tour of an empty lab, but this is maybe the most,
Potential. It's all potential. So
it's all
good.
potential. It's one of the most exciting things that I think has ever happened in my life. To be able to take something that we built out of basic science laboratories and, I don't know, scale something globally. And to be a little bit sappy, that's something that you kind of taught me how to do along the way, and seeing how Phase has evolved as well from a company that I started using when I was a grad student, all the way to now with a laboratory of 20 people.
I'm so old. I'm so old. Yes, it is true. That's part of why we wanted you on here because I mean we started working when you were, like so long ago, and transitioning to faculty and then even spinning off a company too. Like these are all super interesting things, and of course, you know, science being what it is today and all the tumult, a lot of people think about everything from how do I spin up a lab to how do I commercialize? What else do I do with my skills? That kind of stuff. But but let's start sort of at the beginning of the origin story. So like how did we even start working together? Like how did this whole thing get started? Um, and we had something with duckweeds and other stuff. And how did all that evolve into this new initiative you're doing, and your company and all this stuff?
Yeah. So my current laboratory studies, evolution of reproduction. And so I think it's fun to talk about the evolution of, I don't know, kind of our relationship and how, your company grew and how I've been using the technologies in different ways ever since kind of the early days. So I was a grad student at University of Georgia with Jim Liebensmack, working really on large kind of reproductive evolution questions, especially, I don't know, assembling genomes in this kind of weird era of genome assembly where we had all these new technologies coming out and there were new algorithms to put genomes together all the time. And we needed better ways to scaffold genomes. And phage genomics really popped up and started growing when I was a third, fourth, fifth year grad student. And we were using everything at that point, right? Linkage maps, Dovetail Chicago libraries were really interesting at the time. People were using those. And then Hi-C really became accessible. And Phase was really one of those companies that drove the accessibility of that technology. So we started using phase libraries through you guys to generate a handful of plant genomes towards kind of the latter end of my PhD. And then when I started a postdoc at the Donald Danforth Plant Science Center in St. Louis with Blake Myers, um, I started to think differently kind of about accessibility, right? And that's, I think, a really important word to harp on. It was in really the early days of nanopore sequencing when it started to work better. And you could get the starter kit for $1,000. Anyone could plug that into their MacBook and start generating genomic data for genomes of really any size. And I found that to be completely fascinating and empowering I don't know, for a long time, genomics has been sequestered into a handful of core facilities that have ability to spend CapEx, and not everyone can do that. And that means there's this huge gap between people who get to use technologies and people who get to truly understand them, and by touching a technology, it's very different than being an end user hundreds or thousands of miles away. And so I started bringing these nanopore sequencers into classroom. And this was something I was funded by a NSF plant genome postdoc fellowship. And I wanted to be on the academic job market. And so I wanted to build up my teaching skills and really teach creatively. And this idea of bringing a genome sequencer into a classroom, no one was really doing that, so to speak. A handful of people had done that, like CPhages and a few other, very large programs. And I was working on duckweeds as a postdoc. And if you don't know what a duckweed is, it's a really tiny floating aquatic plant, mostly small genomes, 130 megabases for a whole bunch of them. And they are weeds to
Yeah.
most of the world.
We've all seen
Uh,
them. We just don't always know that that's a thing. The little
yeah.
like the little tiny little leafy things floating on top of like swamps and stuff. Yeah.
We'll cut a video into it of duckweed growing, and it's the fastest reproducing plant on the planet. It can clonally divide in as little as 12 to 24 hours, and so this thing is exponentially exploding on the tops of water. From kind of my postdoc perspective, it had all these interesting epigenetic patterns and small RNAs that were just totally missing. Like a lot of genes, 20% of genes were missing relative to like Arabidopsis. So this totally fascinating, rapidly reproducing, some people called it a weed. I called it just this like, reproductive fascination. And so I thought, what a great classroom model that a duckweed could be, because they're invincible. They have great agricultural usage. They can hyperaccumulate nitrogen and phosphorus. They're high protein, so they can be used for plant-based protein alternatives instead of things like pea protein. And so we brought a duckweed into this first year PhD classroom at WashU that I taught. Basically, I had kind of like carte blanche to teach whatever I wanted. So I wanted to do a genome in a box type semester class. And so we brought an nanopore sequencer in, students isolated DNA, basically, 12 first-year PhD students, and we started generating Oxford nanopore data in that class. And right about this time, I sent you maybe a Twitter message that said, "Hey, I'm doing this thing, and I don't really have money, and I think it could be impactful." And you wrote back, that minute and said, "Send me that weird plant." And I remember we shipped it to you, and you generated data.
I remember we didn't even know what it was, and we had a picture of it. We found that it was like covering like a frog. There was like this frog peeking out from water, and there's a bunch of duckweed on it. And we called it frog plant because that's all we knew. That's how we assessed. It's like, what is spirodella? Yeah.
Frog plant. Yep, that's what the Google Drive folder was named with all the data in it that you guys sent us
Mhm.
back. And in that class, these 12 students, which most of whom didn't have a publication yet, most of whom had actually none of whom had ever assembled a genome, most had very little bioinformatics knowledge. Now they had myself and another postdoc, Fiona McLaughlin, who is a mass spec expert. He generated mass spec for that same duckweed accession. So this class had two postdocs teaching them, I don't know, real deal, no BS science of how to do the cutting edge of genomics and genome assembly and comparative genomics. And so Fiona and I got together and said, well, instead of just teaching, what if we produce something? What if these students could be authors on a manuscript that we submit at the end of this semester? And what kind of impact could that have? And I don't know, it worked. And we published a Journal of Experimental Botany paper, JXB paper, of this duckweed genome and the proteome, and discovered some pretty interesting things that, yeah, the proteome is really enriched for photosynthetic genes. The genome is really small and compact, which we kind of already knew, but this Hi-C data that Phase provided allowed us to basically produce the best duckweed genome up to that point. And these students got, in my eyes, an experience that you just don't get in a typical class or even maybe a laboratory experience. And that changed me because I remember asking one of the first year PhD students, how did you get into your dream PhD program? What was it? What was the one thing? And this person said something that I will never forget. They said, "Oh, I got into this program, I think, because I had publications as an undergrad. I had papers because I was working in a lab all 4 years for credit hours, basically. And I was able to put a ton of time in. And it kind of hit me that if you're able to work in a laboratory for credit hours, that means you are not the person that is working in a dining hall for money on college campuses. right? There is this inequity that happens in our field of research, especially at early stages, that disadvantages people from the very, very beginning, even though you might not realize it. And so it started to hit me that what if we could take this word accessibility and new ways of thinking about teaching, and maybe we could level the playing field by giving every undergrad or grad student the opportunity to be an author on a real manuscript, learn these kinds of skills, and I don't know, excel in new ways.
Yeah. Undergrad teaching and just early teaching in the lab is so important. I feel like as far as science goes, for me, for example, I started working in the lab when I was 16 years old. Like there are students, a lot of the science folks, they're driven by just the desire to do science. And the way you inspire that is like a class like yours, right? And then, I mean, nothing is more, I think, inspiring to a student than being like, I discovered a real thing and we put it in a real journal. And it's, like these sort of classes like versus sort of the kind of lecture hall type classes where you're memorizing stuff, which are not, you need those as well, of course, but it's such unique opportunities to really make a huge impact in people's lives. And what you said about, in that, like I remember, working my money job in the day and lab at night, or vice versa, lab
Yeah.
during the day, drive some van or something at night, during the college days. So it's um, trying to help students get that experience, even who normally wouldn't, is like, I think is so transformative in the space.
Completely. And this whole idea of addressing those inequities directly and doing something about it kind of changed me as a scientist. So when I started as a faculty member March 1st of 2020, and probably the most challenging time, maybe other than now, to start a lab and be a scientist. And I remember, that COVID era gave me at least some silver lining time to explore and to think and to ask questions. And I don't know, I moved from St. Louis to Huntsville, Alabama, and I had, in some ways, endless opportunity in front of me at HudsonAlpha. It's a place with access to genome sequencing, always on the edge, agility, all of that. And I was teaching a class at Auburn University, a genomics class. And I remember thinking, well, if Ivan didn't know what a duckweed was, no one really knows what a duckweed was, And like most people don't care. And that's okay that 50 people in the world really, really care about that plant.
I remember you making a joke, possibly during your faculty talks or something, that there is some evidence that it might actually be like weed for ducks because ducks love it.
Yeah.
That's, I was like learning. Yeah.
It's one way to get people to care. Yeah.
But I mean,
And
trying to expand impact of things and getting more people to know about stuff. That was a little bit of a driver for phase too, right? We had this high C tech during the days when putting a genome into chromosomes was virtually impossible. And we had this trick to do it. And we, like when we spun off phase, like I literally spoke to the PIs in charge. I was a postdoc. And I said, we have to turn this into a product or it's going to be a crime against science. And the drive is always, how do we push things forward? How do we make impact into people's lives? How do we make people's lives easier? How do we promote science like to the next, like now, it's almost quaint to think about such a tiny genome as the Spirodella genomes needing like all this genomic, because these days you just zip it together very easily, right? But in those days, it was like a huge accomplishment. And the fact that you did it with undergrads was an even bigger accomplishment. So it was very fun for me to be part of it, in whatever tangential way we were involved.
no, I love that because it was the foundation for something even bigger that has frankly, I think, changed the lives of hundreds and maybe soon to be thousands of people across the planet at this point. So when I started as a faculty member, I was teaching and I was trying to figure out what to do. Like take this idea that you helped me cultivate about like accessible genome sequencing and scaffolding and comparative genomics in a class. And I was thinking, well, what else do you do if not a duckweed? And I remember being on Auburn University's campus, and right at the front gates of the university, beautiful sign. It's right on this corner called Tumors Corner, where a Tumors drugstore is, where you can get milkshakes and stuff. It's like iconic. And there are these two live oak trees that stood guard at the front gates of the university for over 100 years. They're iconic in the sense that after every football game, students would throw toilet paper over them. Students would be taking photos in front of them. They were on every logo of Auburn University. And unfortunately, about 10 years ago, a really fanatic football fan named Harvey Updike, who's now deceased, poisoned those trees in the middle of the night. And no one really figured it out until about a year later when they really started dying. Before they totally died, a handful of biology professors took clonal cuttings of those identical of those original trees, and they kind of just disappeared throughout Auburn, Alabama. So I started asking around, and a biology professor, Dr. Leslie Gertson, who is also the curator of the herbarium on Auburn's campus, said, "I've got one of those. It's in my backyard." And so this idea started to unfold that what if we could build genome sequencing and assembly courses around trees that matter to both students, colleges, communities, alumni, regions, economic regions, it doesn't matter. Something that people can care about. So we launched an Auburn fundraiser called Tiger Giving Day that they do every year. And within 24 hours, we raised $10,000. Alumni came out of the woodwork because it was their tree. And we taught this course over a semester, and students did an oak genome, which is 750 megabases, much bigger. We used high-C sequencing to scaffold it, and they were learning how to use juice box maps and understand how to phase diploid genomes using PacBio hi-fi data and more. And now all of a sudden, more undergrads and grad students are on these publications. And then it grew even bigger because more and more people started hearing about this and asking me, saying, "I want to learn how to use PacBio data. I want to learn how to use Phase Genomics high-C data. How do I do that?" And so I said, what if we could build this into something large? So I wrote an NSF Career Award that got funded by the Plant Genome Program. And the broader impact of that entire grant, the research side was all about exploring sex chromosomes, which we'll talk about later, I'm sure. The broader impact side was about developing accessible genomics education tools to reach the masses. And so I built this program alongside Huiteng Zhang at Washington State University, Les Gertson, Stephen Ficklin at Washington State, called the American Campus Tree Genomes program, ACTG, which is cute and funny. But we basically build Docker containers and curricula for anyone to be able to teach these kinds of campus tree genome classes themselves. So people will go out and find their iconic college campus tree. We even do campus mascots now, if you want to do animals. And we will generate all the data in my laboratory for you. We'll generate Phage Genomics high-C libraries. We'll generate PacBio hi-fi data. We'll generate Illumina RNA-seq or Kinnex long-read RNA-seq. And we'll give you all of that data and the Docker containers and the educational material. And we'll kind of teach the teachers first. And then we'll empower a postdoc or a grad student in that class to be the first author, to like drive all of those students forward and build out their campus tree genome. And so for that, I think we've been able to like democratize access even more, building on top of kind of, your company's ethos to spread it to even more people.
So that's awesome. I didn't know this story was going to have a murder plot in it, tree murder.
Tree
That's
murder.
that's pretty cool. So, there are, scientists do these, and instructors, they want to do these kind of collaborative projects. They want to do these kind of consortia, these kind of large initiatives. But yours stood out because a lot of times they kind of they start and then they kind of fizzle. What do you think was the difference that made yours successful? Like, what advice would you have for folks who are trying to do this kind of thing?
Yeah, I think there's two things. The first is science is better when there's emotion in it, when there's some kind of personal stake. And when you give people the tools to solve a problem in their own community, that changes things. So, for instance, when we started doing these campus tree genome projects at places like Washington State University, where they bred the cosmic crisp apple, WA38, those students who were looking at that cosmic mother tree got to be part of that genome that enriched their state, right? That funded their college. And so there's ownership. And I think that's a key thing. I don't let people in my laboratory, as much as we humanly can, sequence plant genomes that they haven't personally touched or seen or interacted with in some way. That's part of science to me. And I think the second thing that's important to do is um, to give people all of the tools to be successful without having to come back to the well over and over again. So when you build out these huge mega genome projects that we've all been part of in different ways, the communication is always really complex. The infrastructure is complex. And I think the more you can do upfront to solve some of those problems, the better. So between infrastructure, organization, and this kind of emotional personal connection, I think it's easy to empower other people to help you drive large projects forward.
That's cool. And so what do you think is next for this initiative? Like where are you trying to take it next?
So that's a cool question. So we've done 15 campuses at this point. Some really fun ones have been obviously the Auburn oak tree. We did the hedges that line the University of Georgia football stadium. We just did a handful of trees in what's called the McCarty Woods at University of Florida that administrators were talking about potentially tearing down to, build a dining hall or something maybe. And we sequenced those trees, and that effectively, I think, kind of saved them in some way, right? Because now students have a QR code on those trees that said, these students sequenced and assembled this genome as part of a class, like it mattered. Maybe close to your heart, you were at University of Washington, right?
Yep.
With Adam Steinbrenner at UW, we just did the Yoshino cherry trees on
Those
campus.
trees are legendary. Yep.
Legendary. And Adam just sent me some data the other day where they compared it to PopGen data from those trees in Japan and found kind of the closest regional relative for the trees planted and are kind of really understanding where they came from in the transplant process. We started working with someone really interesting that you might know, Dr. Ellie Armstrong. Um, yeah, she's amazing. And we collaborate not just on sex chromosome research, but also on this educational initiative because she started asking, well, Alex, if you're doing trees, why don't you also do animals? And so we started launching the American Campus Mascot program, and that she kind of leads. So we've done a handful of animals, including a beaked whale with Joanna Kelly. We just did a handful of birds with some other groups, and Ellie did a gray fox. So we're expanding kind of into new kingdoms of life. But really, I think the bigger thing that we can do is I looked at things like LinkedIn Learning. I look at Coursera. I look at all of these ways that people are learning the next generation of genomics and bioinformatics and computer science and AI. And I think someone like me and you, we're at, the interface of the technologies. So what could we be doing to create these kind of large scale, democratized, interactive courses where people are touching the very latest generation of data? Not taking classes on microarrays and things like that. I even did that as a grad student. It wasn't even relevant to me necessarily. So how do we take your technologies and I don't know, put it on the internet for global distribution?
Yeah. I mean, that's always been the challenge with genomics, right? Because it requires so much sort of academic knowledge and you have to be assigned it. But as it's matured over the years, the decades now, it's becoming more and more household, right? It's becoming a little bit more accessible, a little bit easier. People like I remember when we first started, um, how our technology worked and all this kind of stuff, people would use it and then they would give presentations at conferences. And there was always a slide or two where they would explain how the technology works. They're like, okay, here's the flow chart. It does this, 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 this. And I remember like 3 or 4 or 5 years in, there would be a whole session where basically every talk would have something that they assembled, with our stuff and they would just skip it over. They would just go like, we did it. We used like PacBio long reads or nanopore long reads. We used Hi-C. We scaffolded it. You all know how it works already. So I'm just going to talk about biology. And so the idea of like moving from explaining the technical details of how the thing works to specialists and instead just going, let's just talk about the biology now. That's now, like you drive a car and you don't need like a university degree on how to build cars. They just work, right? So moving it towards something that's more applicable to a layman audience, that goes right into kind of um, sort of like useful insights and understanding without having to go into the nerd out on the technical details um, is super cool. And it's just an important part of the evolution of every technology, right? Like just got to become
Totally.
easier and more accessible to people. And I think, you're doing such a great job at sort of promoting that and driving it in that direction. Um, Let me take a little bit of, so we talked about this, you mentioned this a little bit earlier in the talk. Let me ask you this. So postdoc, faculty now, and then of course now your transition to entrepreneur. Let's talk a little bit about that, right? So What made you, because you have this awesome initiative, you're doing all this cool research, you're at HudsonAlpha, which is an amazing place, right? What made you want to spin off a company? Right? Like, what drove you? What's your vision for it, etc.?
When I was a postdoc at the Danforth Center, there was this really amazing program, um, Arch Grants and BioSTL and BioGenerator in that community. And there were all these startup communities all around St. Louis. And there was an entrepreneur in residence at the Danforth Center who would effectively walk around the building and sit down in our labs and say, "What are you doing that's interesting? What do you got? What's in your brain?" And it's so funny because as academics, we're conditioned in a lot of ways to be giving away our discoveries, if you want to think of it that way, in terms of publication. And I think that's one of the greatest gifts that we as scientists can give to the world, truly. It is why we do what we do, is to disseminate knowledge for the greater good. I also think there's a lot of very valuable ideas that you could capitalize on and still meet that goal of putting technologies out into the world for a greater good. So I always wanted to do that. And I was always kind of trying to find the right thing. I worked on sex chromosomes. I'm 36 now. I started working on them when I was 18. So half my life I've been thinking about XY, ZW, and UV sex chromosomes across plants and animals. And I always really was obsessed with this idea that if you could control the flower in some way, if you could make sex chromosomes, engineer them, that you could control food. You could control the way that we breed, control the way that we eat, right? In some sense, every single thing that we eat comes from a flower, right? Comes from the act of sexual or even clonal reproduction. And so I was obsessed as a postdoc with maybe building out a company, building out technology, thinking about developing IP around engineering new sex chromosomes. And it just was tough. I was trying to get a job and all of it, and it just didn't come to fruition yet. So when I started Hudson Alpha, I'm around all of these new technologies. I'm touching PacBio sequencers. We've got NovaSeq in the laboratory. We've got 10x single cell machines, just around the corner from us. And I'm thinking like people come to a place like Hudson Alpha because they want to generate ultra high quality genomic data quickly to get to their answer. And that was like a kind of revelation for me in some ways. My background is evolutionary biology. I was always thinking about the past. And I find that very enriching, to think about hundreds of millions or even a billion years of time that green things have been evolving. But then, I meet a faculty member, Josh Clevenger. We actually overlapped during our PhDs at University of Georgia. He started as a faculty member about the same time as I did. And he's a plant breeder by training and bred new varieties of peanuts at University of Georgia. He worked at Mars Corporation, kind of leading a lot of these large peanut improvement initiatives. Um, and so he made me start thinking about, I don't know, how can you use genomics to change the future? Which was kind of a big pivot for me in a bunch of ways. And I started to see what he was doing in his research program, which was to develop genotyping at scale using low pass Illumina sequencing. He was basically doing tens of thousands of libraries and genotype calls per year, supporting, I don't even know how many countries at this point, maybe 20 countries worth of breeding programs. And I started to see what one person in a laboratory could do to have impact on the global food supply. And at the same time, Josh and I were, always trying to be on the edge of long read sequencing. And so, I had kind of my expertise in PacBio from the early days. I've been using them since the RS2 when we sequenced the asparagus genome back in 2013, 2014, which took 5 years and $1 million and a huge international collaboration. And now we're at this place where we have a PacBio Revio in our laboratory, just the two of us, that we run for our research program and for fee for service. And so we started to explore the idea of, I don't know, doing things differently, which is kind of a benefit of what you can do at a place like this. So instead of sequencing one genome on one PacBio Revio cell, we started to ask, what if you sequence 12, 24? What if you put 96 libraries on a single Revio cell? You generate less data per individual, but you can call SNPs, indels, and structural variants all in a single read. What if you could change the way we think about complex genotyping and deliver answers to people faster, to in some ways use genomics to change the future? And so that's really how Veil started and how this kind of journey of entrepreneurship began, a bit of an evolution, just like you talked about with Phase. that when you are on the edge and you understand the technologies and you understand the market that could use it, I think that's really where the sweet spot comes in. And I've never started a company before this, but we are surrounded by this entrepreneurship ecosystem here. There's 55 companies in this building. And so we've got, no dearth of people that are helping coach us kind of all along the way.
That's super cool. What would be what would be a piece of advice from you for someone who is maybe faculty? Well, you're faculty going, so right, so I coach like postdoc startups and all this kind of stuff all the time, so I have a lot of thoughts on the subject, but specifically from a professor's perspective. Professor on the cusp of going into entrepreneurship, what how would you advise someone who is in that place, who hasn't really made the transition yet?
Yeah, I think a lot of people have a lot of really good ideas. And I think even sometimes a mediocre idea can be a very, very successful company. The market has to be in the right place at the right time to accept your great idea for it to have impact or meaning. I think a lot of really great ideas end up on kind of the shelf of your brain. You and I have probably had a bunch of those. A lot of people probably have. And so there's the hard part. I think Sidney Brenner, I never got to meet him, but he was totally renowned for giving away great ideas all the time. And he would always talk to people and offer advice and offer interesting conversation about changing their ideas and whatnot. And he had no ego about it. Because I think a good idea is just the starting place for these kinds of things. And so what I've really appreciated, and really something that kind of you said at the beginning, I think your network is the most valuable thing that you have as a scientist. Because at the end of the day, just about every scientist I meet is smart in some way. Maybe not smart in the same way that I am, or the same way you are, but everyone has some kind of niche or skill. The hard part is understanding how you connect multiple people's niches and skills together to elevate your great idea to the next level.
Yeah. Yeah, I agree. I would add one little spice, like maybe a little piece to it. Is that entrepreneurship is really about action, and that, like what you said, even mediocre ideas can make good companies, but a great idea without action will never make good company, right? So like, it's not about who's the best scientist, it's about pushing something forward and like making it go. It's really, that's what sometimes they call it grit, sometimes they call it
Yeah.
initiative, whatever it is, but it's this desire and need to constantly push, push, push. And
Yeah.
so that's really it. I mean, because the issue is like a lot of times professors, you have a faculty job, you're a professor. Like you don't need to kind of go on a limb and risk your career credit to start these to do these things. But it's this kind of internal drive. It's this like fire in your belly to like you have to do more, right? You have to like make more impact. And so just kind of try to enable that by surrounding yourself with people who kind of can help you where do things that you don't know how to do. Yeah. No, it's super cool. Um, Alex, I let's do something fun. Let me ask you some fun questions. We've been talking for a while, and I think we've established you're great at science. What what are give me some of your favorite um like scientific guilty pleasures that maybe not everyone has.
I definitely like stealing all of the NAB ice packs out of the enzyme orders and bringing them back
Mm.
to my home fridge. And I have really no use for them most of the time, but I just like having a freezer just filled with those things. I love going in and getting the bubble wrap and stomping on it before people get in the laboratory. Um, and my grad students and postdocs don't know this, but I love going and reading everybody's lab notebooks after everyone has gone home.
Oh, that's a very guilty pleasure.
It's like reading someone's
That might,
diary,
I feel like there might be something in the Geneva Convention about not doing that. Uh, cool. Well, listen, man, this has been fun. I'm glad we got to talk. I've enjoyed working with you for years. I'm glad, like, again, like we were talking about how it's so cool to be able to make impact like this. It was the same thing for us, right? We had this quirky little sequencing tech. We said this can help a lot of people. And the first time you see somebody take the thing you made and use it for something, even like without you, when somebody uses one of our products and never talks to us again and just paper comes out, that's like insanely rewarding. Like the idea that you made a thing and somebody else benefited from the thing, it's like it's something that one of the benefits of being a scientist and scientist entrepreneur is. If people want to learn more and they want to talk to you, how do they find you? How do they learn more about what you're doing?
So from the academic side, if anyone is working on sex chromosomes or reproductive biology, of which IC sequencing has been foundational to our ability to explore across the tree of life, you can find me on the HudsonAlpha faculty web pages. If you're interested in long-read low-pass sequencing at scale, you can find me at veilgenomics.com. And if you're interested in the American Campus Tree Genomes program, it's also on the hudsonalpha.org/ACTG. And feel free to reach out to me anytime about genomics, sequencing, sex chromosomes, accessible genomics, and entrepreneurship.
Very cool. What else do you want to plug? Anything else before we go?
I think that's it. Honestly, the ability to do high-C sequencing at scale has changed the direction of my research career. We've sequenced hundreds of high-C libraries using kits like yours and with you guys as a commercial vendor. We've published 50 to 100 genomes at this point. We've sequenced hundreds of Arabidopsis high-C libraries to explore climate change questions related to how CO2 affects chromatin structure. And so I just think your company has empowered a lot of people to do excellent science. And for that, I'm appreciative of you.
Well, thank you so much. I'm blushing over here. Well, Alex, thank you. And to the rest of the folks watching this, if you want to learn more about Phase, follow us on the various socials. Check out our website, phasegenomics.com, and just shoot us an email. We love talking about science. Everything's not going to be a sale. We're just, a company of scientists trying to do good science. So, thanks everyone, and that's it.